GUB Full Sample Processing. Uncap the tubes and label one aliquot with the MRN number. Obtain the lavender top EDTA plasma tube and collect the plasma into one cryo vial using the P1000 pipette and 1 milliliter filter tips, and make sure to have enough volume for further aliquots into the appropriate number of cryo vials. Obtain the golden top serum tube and use the P1000 pipette with the 1 milliliter filter tips to aspirate off the serum and aliquot equally into the cryo vials. When removing the serum, take care not to disturb the gel layer or the lower red blood cell layer. Obtain 2 round bottom tubes and obtain the EDTA plasma once again and use a transfer pipette to aspirate off and discard the remaining plasma into a 5 milliliter round bottom tube down to 1 millimeter from the red blood cell layer. When removing the plasma, take care not to disturb the white blood cell layer, also called the buffy layer, which forms a thin film between the upper plasma layer and the lower layer of the packed red blood cells. Samples with exceptionally high white blood cell count will have a thicker buffy coat. Using the same transfer pipette to carefully aspirate the exposed white blood cell layer. Aspirate slowly using a circular motion to pull all the visible buffy coat material into the transfer pipette. Some contamination of the white blood cell layer with the underlying red blood cell layer is expected. So, using the same transfer pipette to carefully aspirate the exposed white blood cell layer, aspirate slowly using a circular motion to pull off all the visible buffy coat material into the transfer pipette. Some contamination of the white blood cell layer with the underlying red blood cell layer is expected. Collect this buffy coat into one cryo vial and aim to make equal volumes of aliquots with the P1000 pipette and 1 milliliter filter tips before storage. Discard the excess plasma in the biohazardous waste. Obtain the urine tube and collect 2 milliliters of the urine supernatant aliquots into the appropriate number of cryo vials. Avoid pipetting near the bottom of the centrifuge tube to maintain separation of the urine and the urine pellet. Decant the excess urine into another tube, but be sure to leave 2 milliliters of urine to resuspend the pellet. Resuspend the pellet in the urine using the pipette. Collect 1 aliquot of the urine pellet into the cryo vial. Collect 1 aliquot of the urine pellet into the cryo vial. Record the volumes of the Buffy, serum, and urine collected on the back of the MRN label. Record the volumes of the Buffy, serum, and urine pellet and write it on the back of the labels. Recap the epitubes with the purple lids on the plasma, the yellow lids on the serum, the pink lid on the urine pellet, and the clear lids on the urine. Label the epitubes with the appropriate label.